The older technique is a technique called Sanger sequencing. That was sort of the one of the earliest ways of sequencing DNA and understanding what the actual sequence was. And when we first started learning about the importance of specific genetic mutations in AML, the tests that we sent for patients were all based on Sanger sequencing. The advantages of Sanger are that it is quite accurate, and because we're only assessing a small amount of the genome, it's there are these single gene tests. The individual tests are cheap. The disadvantage is they are single gene tests, and so to actually assess a large number of genes, you would have to send many different individual tests. Those actually become quite costly. So even though an NGS panel is more expensive than a single Sanger test, it's actually much more cost advantageous to do next generation sequencing. It's also become faster, and you get a lot more information with it. So while there are a couple of specific genes that still benefit from being sequenced the old way, by and large, the need for faster turnaround time, higher throughput, and being able to get information about many different genes at once has meant that I think mostly NGS platforms have greater advantages.